Good morning. Today we have to study chromosomal theory of inheritance. Earlier we have discussed about the Sutton and Bavary chromosomal theory of inheritance. Today, the T.S. Morgan, Thomas Hunt Morgan is the name of scientist who experimented or experimentally verified the chromosomal theory of inheritance. He worked on the chromosomal theory of inheritance in the form of experimental verification to evaluate that how does variation arises due to sexual reproduction. For his experiment, he selected tiny fruit flies named as Drosophila melanogaster. Why he selected Drosophila melanogaster only? There are many reasons of the selection of Drosophila melanogaster. One is it can be grown on simple synthetic medium in laboratory. In the laboratory, not in the garden, not in the agricultural field, anybody will have to wait. It can be easily grown on the simple synthetic medium in laboratory. Second point, their life cycle completes in about 2 weeks. A scientist will have to not wait for more time. In 2 weeks, the scientist can get the next offspring and can compare the set of characteristics of the parental combination to their next offsprings. Third is, their single mating can produce a large number of progeny flies. Means what the single mating? The scientist will have the imagination that if the one sexual intercourse, one mating is going on between the male and female flies, then many offsprings will be growing. And he can compare out of these many offsprings what a set of characters have been transferred to particular group of the offsprings. Particular set of the offsprings have what have the characters. What the new characters have arises. What the parental characters have not arises in this particular generation. So all these comparisons can be established. All these comparisons can be established between the parental combination and the successive generations of offsprings. Next one, there is very clear differentiation in male and female body. If we can compare the male and female body, anybody can easily distinguish between the male sex flies and female flies. These are easily detectable. Last one is that it has many types of hereditary variations that can be seen with low power microscope. Many types of hereditary variations means what type of hereditary variations, what type of characters which are exhibited by the parental bodies, and what type of characters have been transferred and inherited into the next offsprings can easily be seen by the low power microscope, which is easily available to us. So these are the causes why Drosophila melanogaster was selected by Thomas Hunt Morgan for the experimental verification on the chromosomal theory of inheritance. In the next subtopic of the chromosomal theory of inheritance, we will have to discuss the two subtopics. One is the linkage and second is the recombination. Linkage, we know that linkage is the process of physical association of two genes on the same chromosomal arm. And these two genes are tightly linked in such a manner that they do not segregate from generation to generation and retain their physical association from one generation to next generation successively. So that phenomena of the physical association of two genes on the same arm and which are getting transferred from generation to generation without getting segregated is known as linkage. But we will have to study about the recombination is the process, as the term is saying. Recombination means combination has been repeated, combination has been changed. Means now whatever the set of characters were existing in the parental generation has now changed, has now recombined, has the new recombinant, have the new set of characters can be seen in the next offsprings. The process of arrival of new combination of genes after the exchange of gametes during the crossing over during the meiotic cell division in the next offspring is known as recombination. is known as recombination. And we know that Morgan hybridized yellow-bodied, white-eyed females Drosophila melanogaster with brown-bodied, red-eyed males, and found that. And when he got the F1 generation, in the F1 generation he found that two types of organisms were obtained: yellow-bodied, white-eyed, and wild-type animals were obtained in the F1 generation. And when he established the selfing intercrossed, he found that the F2 generation, what the Mendel had said during the dihybrid cross, 9 is to 3 is to 3 is to 1, the phenotypic ratio will come, that was not coming in the second generation. So this was a deviation from the dihybrid cross obtained in the form of phenotypic ratio by the Gregor von Mendel. So this is why he verified it, why this is happening, why the 9 is to 3 is to 3 is to 1 is not coming, which is the result of independent assortment. Why that is not coming? Then here the answer was given that because that was the linkage was there, and that some genes were tightly linked and had not segregated from generation to generation. That's why, due to not allelic independent assortment, due to the tight physical association of the genes, now the 9 is to 3 is to 3 is to 1 have not arrived in the next generation. Second, he found that the proportion of parental gene combination were much higher than the non-parental combination. This is about the linkage. In the linkage, it has been seen that due to the absence of segregation in the parental genetic combinations which were arranged on the same chromosome, it was found that the parental combination were obtained in the successive generation, and that generation it has been shown in the NCERT figure in the page number 84 in the figure 5.11 that recombination has taken place only 1.3% in yellow-bodied white-eyed females and brown-bodied red-eyed males, and parental combination have come 98.7% in the successive generation. This is showing that this is the linkage, this is the tight physical association of the genes which have not segregated in the successive generations. But in the second figure, in the 84 page number, in figure 5.11, we have seen that the parental combination, due to the loose arrangement of the genes on the same chromosomal arm, it has been seen that the genetic exchange, the chromosomal exchange has happened during the chiasmata formation, after the chiasmata formation during the meiotic division, that recombinants have come 37.2%. And why this 37.2% new recombinants have arrived in the next offsprings? Due to the loose combination of the genes. And parental combination have remained only 62.8%. So this have been crossed between the white miniature bodied and wild type Drosophila melanogaster, and it has been seen that parental combination have reduced to 62.8%, and the recombinants have come to 37.2%. This is saying about the loose combination of the genes on the chromosomal arm. One student of the T.S. Morgan, who is Alfred Stewart, and he said that frequency of recombination between gene pairs on the same chromosome as a measure of distance between genes means genetic mapping. Alfred Stewart has measured gene mapping and the distance of the two genes on the chromosomal arm. This was the role of the Alfred Stewart, who was the student of Morgan. Thank you.